Thank you very much, Barbara. Thank you very much for this kind invitation. And it's a pity that I can only visit all of you guys virtually, but I still am enjoying a lot my visit. So today I'm going to talk about a new topic in the lab. So we have been working on this topic only for the last 5 years. And just to give you a brief introduction about some of the projects that we work on, and Barbara already mentioned many of them. We work on sex chromosome evolution in vertebrates, mostly in primates. Recently, we have been the members of the telomere to telomere consortium to sequence the Y chromosome in human, but also in other primates. We work on mitochondrial DNA evolution in mammals. We also have a more applied project on childhood obesity. And the project I'm going to talk about today is on non-BDNA. You can ask, what do these all projects have in common? And they have my interest in mutations in common. So I worked on mutations for my PhD. So for my PhD, I studied the genetic consequences of Chernobyl accident in Ukraine, identifying mutations in small rodents that we were catching there. And since then, I kept my interest in studying mutations. So before I talk about non-BDNA, I just wanted to remind you what a BDNA is, and that's something that you are all familiar with. So this is the conformation of DNA that was discovered by Hudson and Crick, and it is a right-handed helix with about 10 base pairs per turn. It's called BDNA because also some other conformations were discovered, like aDNA, depending on the presence of salt in the solution. So, but while everybody is familiar with the Hudson and Crick structure, not every biologist even knows that about 13% of the genome can fold into other conformations. And here I'm listing some of these conformations. This usually happens at certain motifs. For example, if there are inverted repeats, they can form cruciforms. Mirror repeats can form hDNA, which actually is a triple helix, not a double helix, but a triple helix. Direct repeats can form slip strand structures. Also, microsatellites is a version of direct repeats with the slip strand structures. Then there are also G quadruplex motifs, Z-DNA motifs, and A-phased repeats, and I'll go through some of them in a little more detail. So perhaps the most studied non-BDNA structure are G quadruplexes. And if in the genome there are motifs that include at least 4 stretches of 3 Gs in a row, then these Gs can pair with themselves into these plenary structures. So let's follow this structure here. So this is DNA strand, 3 Gs. Then there are some other nucleotides, again 3 Gs, some other nucleotides, 3 Gs, some other nucleotides, and 3 Gs again. Then within each of these planes, there is hydrogen bond formation among different guanines, okay? And this structure can be quite strong, but it is still not, it is maintained by hydrogen bonds, and it is stabilized by cations. So it is still transient. So this is a more detailed illustration of another non-BDNA structure, and this are the cruciforms. So if you have inverted repeats, So this is what we have on the Y chromosome, right? So in the palindromes, they can form cruciform structures in which the arms are paired. And direct repeats can form slipped strands, right? Because pairing can occur between repeats, different copies of repeats. Okay, so here, this is what's happening. The different copies of repeats are paired. The first one from the upper strand with the second one from the upper strand with the first one from the second strand. And then there is pairing leading to loops, and we call this slipped strands. Mirror repeats can form DNA triplices, okay, in which there is formation of the triplex structure. And another type of non-B DNA are A-phase repeats. So this is in a way somewhat similar to G quadruplices, but this is a structure that is based on adenines. So if there are several adenines in a row followed by some other nucleotides, which we call them a spacer, then there is increased DNA bending at the stretches, between stretches of A's, leading to A-tract bend structure. And the last non-B DNA structure that I'm going to talk about is zDNA. So zDNA forms from purine pyrimidine repeats. And in fact, it is a left-handed structure that is more loose compared to a B-DNA. So, for example, it is more susceptible to radiation damage. So I hope got the flavor of what non-B DNA is. And one can think, okay, so what? Non-B DNA, it exists, but I'm going to convince you that it doesn't only exist in the genome, but that it is important. So first of all, it plays important roles in the healthy genome, mostly focusing on human right now, although it is actually present in almost any species scientists have looked at. So G quadruplices, they form at telomeres. So remember, what is the consensus repeat for telomeres? T, T, A, G, 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 repeated multiple times. So you have 3 Gs in a row separated by some other nucleotides. These Gs, they can pair among themselves on the same strand, right? And this is what protects telomeres, one of the mechanisms of telomere protection. Also, G quadruplices are enriched in origins of replication in mammalian genomes. Not all origins of replication have G quadruplices, but about 85% of origins have them. And there they are functional. They are contributing to the replication initiation. The same is true for promoters. Many promoters in mammals have G quadruplices in them. And you can imagine that if the structure forms at one of the strands, it helps in strand separation, which is what's needed during transcription initiation. Another function of G quadruplices is that they participate in the life cycle of some transposable elements. And a prominent example are L1 elements. And this is the work that was published in 2017 from another group. There is a G tract right here, a G quadruplex motif. And when it was deleted in experiments, the transposition, retrotransposition activity of L1s decreased substantially. This suggests that they are important for the retrotransposition activity. Some other functions of non-B DNA HDNA, A-phase repeats, zDNA. These structures, they regulate transcription and translation. And there is now genome-wide evidence that they form in the cells and they participate in regulation of many different cellular processes. And why are they such good regulators? And this is because their formation is transient. They're not always forming the structures. These motifs, they can also form regular bDNA, but they can be used as sensors of environment in which they can participate then as regulators of multiple processes. Non-bDNA also plays important roles in disease genomes. Non-bDNA is associated with chromosomal rearrangements in cancer. So these unusual structures, they are more fragile, they're easier to break, especially in cancer cells. Then non-bDNA is formed at trinucleotide repeats, and expansions of these repeats are associated with many neurological and muscular degenerative diseases. And then another example, which I really like, is shown here, is that G quadruplices in particular are formed at the hexanucleotide repeat expansions at the locus of C9orf72. And as a result, they are a major genetic cause of amyotrophic lateral sclerosis. So this is how they form in DNA. But then sometimes they can still be transcribed into RNA, okay? And these transcripts, then they lead as magnets of accumulating of nucleolar proteins and accumulation of toxic proteins, mostly within the nucleus, but then also of abortive transcripts that lead to accumulation of stress proteins in the cytoplasm. So it is repeat located within an intron. The normal copy number is from 2 to 8, but in the disease it's hundreds to thousands. So the cell becomes overwhelmed with these stress proteins that result from the translation of G quadruplices. So I hope I was able to convince you that they are important. But I always think about non-bDNA as a blessing and a curse of the genome. So I just mentioned to you that they play important roles, that they protect telomeres, they participate in regulation of gene expression by opening up promoters. I also listed some other functions. They are important for origins of replication. But let's think about it also this way. Imagine that there is a polymerase trying to replicate through non-bDNA. Then when it encounters this structure, it's possible that polymerase can pause or stop or introduce more errors. And this is where the curse comes in, that non-bDNA can impede replication, can interfere with replication. And as I already mentioned, for cancer genomes, non-bDNA can lead to increased chromosomal instability. So what does all of this mean? Is that non-bDNA can be an important driver of mutations if indeed polymerases have more mutations at non-bDNA. Also diversity, they can contribute to adaptation, but also, as I mentioned, non-bDNA is associated with neurological diseases and cancer. I talked about non-bDNA being transient in the genome, and formation of many non-bDNA structures is regulated by the presence of certain ions. And this is particularly important for plants. So if there is increased concentration of potassium, then the G quadruplices will be forming more easily. But the increased concentration of potassium might be occurring under conditions of drought. And these consequences for non-BDNA formation are particularly important under the current climate change and global warming. So the applications of non-BDNA are multiple for disease, for agriculture, but also for evolution, and that's why my lab has become interested in it. So today I'm going to talk about some of our recent studies about non-BDNA. I will talk about polymerization kinetics and accuracy at non-BDNA in vitro. And then I'll talk about the contribution of non-BDNA to mutation rates in vivo, about selection at non-BDNA, and I'll talk maybe a little bit about success of major sequencing technologies at non-BDNA. So moving on to the first topic, we wanted to ask a question, is whether non-BDNA affects genome-wide polymerization kinetics and accuracy in vitro. And it sounds like, okay, so what does this question have to do with anything? But let me tell you a story. So when we were working on sequencing the gorilla Y chromosome, we did this in 2016, and we used PacBio sequencing, which was in the stage of development. We noticed that in addition to information about the nucleotides, deciphered nucleotides, PacBio sequencing was also giving us information about the timing of incorporation of these nucleotides. So because PacBio sequencing is based on polymerization, it also gives information about how quickly these nucleotides are incorporated in the sequence that is being deciphered. So it gives information on this interpulse durations, or IPDs. So the single molecular real-time sequencing, this is SMRT sequencing or PacBio. It is strand-specific, and it uses a polymerase. In fact, it uses a modified bacteriophage polymerase. And when we just did a quick analysis of this interpulse durations of how the timing of incorporation of nucleotides, we found some irregularities. We found that sometimes this IPDs, or the timing of incorporation, is affected by non-BDNA motifs, okay, by this stretches of Gs for G quadruplices and so on. So this prompted us to design a study about how sequencing, timing, and also sequencing accuracy within the PacBio instrument is affected by potential presence of non-BDNA structures. So for this, we used the annotations of non-BDNA motifs from the non-BDNA database. And these are the types of non-BDNA motifs that we've used. This includes A-phase repeats, direct, inverted, mirror repeats, G quadruplex motifs, ZDNA motifs, as well as microsatellites that we annotated with our software STRFM. So all of these are the motifs that I was talking to you about already. And then we used the publicly available sequencing data set. From the Genome in a Bottle Consortium. So this was in 2016 when this data set was released, and a single individual human was sequenced with PacBio technology at 69x. So this is still a very large data set. It's still used in many genomic studies. So we took advantage of this publicly available data sets, completely publicly available data sets. And how does the PacBio SMRT sequencing works? So let me just walk you through this. And at the core of PacBio sequencing is that it is sequencing from a double-stranded template. And here, this double-stranded template is in the middle, okay? And it also has the adapters attached to it. So here I'm showing the double-stranded template, which has this bell-shaped adapter structure, or you can also think about it as a bone that you're given to your dog right in the middle here. And this is a toy example, and I just put zDNA on it, CTG repeat, a microsatellite direct repeat, and on the other strand, there is also zDNA, right? There is an AGC repeat. There is also a direct repeat. G quadruplex is present only on one of the two strands. But then what happens is that DNA polymerase during sequencing, PacBio sequencing, goes around this template, and sequences both strands. It incorporates nucleotides based on this template, this double-stranded template, and goes through this multiple times, generating almost like a serpentine, right? A snake-like long sequence that has many subreads. And at the same time, it generates the data on the timing of incorporation of these nucleotides. And now I will just walk you through, okay, we're in this serpentine, we're still going around and around with the polymerase. So bear with me, this is the end of this snake-like structure. And so what we did is we combined the data on the timing of incorporation of nucleotides from PacBio instrument for the human genome. And we combine the data, so here is just the data of IPD from multiple subreads for this template, right? But then also subreads from other templates for G quadruplices, for zDNA. And we just combined all of this data, and we grouped this data by motifs. So we took the data from all G quadruplices in the genome. We combined all of this data for IPDs, for the timing of incorporation of the nucleotides, and we got a distribution. So this is the distribution for the timing of incorporation of nucleotides at G quadruplices. We did the same for zDNA, for direct repeats, and for many different types of microsatellites. But I'm just illustrating here the G quadruplices. And we also generated controls. So these were sequences that were free from any non-BA motifs, and they're shown here in blue. And then our interest was to compare the two distributions, right? Between the motifs distribution for timing of incorporation nucleotides or for motifs versus for the controls. And we used sophisticated statistical techniques of interwise testing to compare these two distributions. Yeah, so, and what we found is that here it's the same idea, right? It's just following the same colors just to keep your intuition. We're comparing these two distributions. Now this is not an example anymore. This is real data. So red distribution in red is for G quadruplices, and in blue it's control. And you can see that the distribution of the timing of incorporation of nucleotides for G quadruplices is significantly different than it is for control. In fact, the timing of incorporation for G quadruplets, when it's used as a template, it is slower, right? So polymerase slows down when it goes through a G quadruplex. It's not something that we observe on the other strand. There is some noise, but we don't have this nice and consistent pattern. So if there is a row of C's on the other strand, we don't see the polymerization slow down. We also observed some interesting patterns of polymerization profiles at microsatellites, suggesting some periodicity. And this polymerization changes, they might be important for driving some of the diseases. So the CAG repeat shown here, when it's expanded, it contributes to Huntington's disease and to spinocerebellar ataxia. So as a result, what we were able to do is we could combine the data on polymerization speed across many different non-B motifs, also many microsatellites. So this is a genome-wide data set in humans, and red indicates slowdown and blue indicates speed up. Some motifs were leading to speed ups in polymerization speed, and G4s, G quadruplexes led to very significant slowdown. And then some other motifs, they were changing speed in more periodic ways. One thing that we've noticed here is that a GGT microsatellite, even though it is not a canonical G quadruplex, right? Because for G quadruplex, you need to have at least 3 Gs in a row separated by some other nucleotides. But the profile of polymerization speed reminds of a G quadruplex for this microsatellite, suggesting that perhaps GGT repeat forms a G quadruplex. And this is exactly what we observed in our wet lab experiments. And in general, what we could also do is we could correlate the IPD values, the speed of polymerization, with the biophysical properties of G quadruplexes. So here I'm finding the relationship, the correlation between average IPD values and the circular dichroism intramolecular structure, the delta epsilon. So it's a highly significant relationship, suggesting that G quadruplexes indeed might form within the PacBio instrument. So the other question that we asked is whether non-B DNA likely forming within the instrument can affect also the sequencing error rates. And we are finding that G quadruplexes in particular lead to increased mismatch errors within PacBio sequencing. So the incidence of mismatches is increased about twofold at G quadruplexes as compared to other sequences. There are also additional effects. For example, G quadruplexes increased deletions for smart sequencing, but these effects are smaller. So, and we also are finding that the incidence of errors is correlated with the incidence of slowdown, with the slowdown of polymerization, particularly for G quadruplexes when they're used as a template. This is a much stronger relationship than what we are finding for the other strands when the C-rich sequence is being sequenced or for controls. So we have this hypothesis that polymerase slows down and makes more errors when it goes through a G quadruplex. And just to summarize this result, this is a cartoon figure by Wilfried, who did most of this work, a former graduate student of mine. So when polymerase encounters a G quadruplex, it makes more errors and its speed goes down. Okay. And if you're interested more in this work, it was published in Genome Research several years ago. So, of course, this is great. This is exciting, interesting, but this all is happening within a sequencing instrument. So it has important consequences if we want to sequence non-B DNA accurately, but we were thinking whether the same might be happening in living cells. And the question that we wanted to ask is, does non-B DNA contribute to regional variation in mutation rates at the small scale and or at the large scale? So for this, we used some other data sets, and we used the polymorphism data for humans for the Simon's Genome Diversity Project. So this included 44 million SNPs from 279 individuals. This was sequenced at 30x read depth. And we also used the divergence or fixed differences. Divergence was from human orangutan comparisons. So this included 69 million mismatches between human and orangutan. So the first thing that we did is we detected the small scale variation of nucleotide substitutions. So we examined 6 types of non-B DNA and separately, and then we compared the distribution of divergence to diversity there, genome-wide, right? We combined all of this loci against controls. And we scaled loci, meaning because they are obviously of different length, and we found the differences with the same functional data analysis techniques that I mentioned to you previously that we used in our IPD project. So let me show just some of the results. So here in blue is what we observing for the inverted repeats, and in black there is a control. So for inverted repeats, we are seeing elevated SNP frequency in spacers, but actually lower SNP density in repeats, suggesting that inverted repeats might be driving the elevated mutation rate. But in repeats themselves, there could be gene conversion homogenizing SNPs there. And within G quadruplices, we could find the elevated SNP frequency both in the stretches of Gs, we call them stems, but also in loops. These are the nucleotides between stretches of Gs. So there is increased substitution frequency within G4 loci, and this was confirmed by our analysis of divergence. So the other thing that we did is that we can use this software Quadron that can predict the structure of G quadruplices and can also predict its stability. So it divides all G quadruplices into stable versus unstable. So what we are finding is that the substitution frequencies, the SNP frequencies in this case, are higher for stable G quadruplices, for stronger structures, than for unstable G quadruplices. So this is something that also suggests that the structures are driving the increased mutagenesis at this loci. And the last thing that we did in this project is we looked at the large-scale regional variation in substitution rates. So the previous slides just looked at the small scale, right? So this like 1 to 30 nucleotides for stems and 1 to 20 nucleotides for loops. And also we looked at plus minus 100 base pairs. But here we looked at the 1 megabase windows, and we studied how diversity or the SNP frequency and divergence depend on the presence of non-B DNA and some other factors that we knew from previous studies are important, such as replication timing, recombination rate, epigenetic modifications, and so on. And what we are finding is that non-B DNA contributes to large-scale regional variation in substitution rates quite substantially, okay? So the R squared of our models, the percentage of variability explained is quite high. It's about 56 to 58%. And the non-B DNA can explain 16 to 20% of variability in these models, which is quite substantial. So it is frequently, in these models, it is the most important predictor. So to summarize this part of my presentation, if there is a non-B DNA present in the genome, it usually increases the nucleotide substitution frequencies. So indeed, most likely the structures, they function as mutational hotspots. And this work was published in Nucleic Acids Research last year, so you're welcome to go to this publication for more details. So this conclusion has really important consequences. And I'm going to illustrate one example. And this is a paper that was published a couple of years ago in Science. And it has to do with the importance of non-B DNA for parallel evolution in fish. So in stickleback fish, there are two populations, marine populations and freshwater populations. And morphologically, they differ in the presence absence of dorsal spines and spiny pelvic fins. So freshwater populations, they lack those spines and this fin because there is no need for them to fight against the waves. They don't use the spines and this spiny fin at all. But so they can conserve the energy on producing these structures. But what is the molecular mechanism behind this? And this paper goes in a lot of detail. So the paper by Shi and colleagues from 2019 goes into a lot of detail about how this is achieved at the molecular level. So there is a gene which is called PTX1 gene that is important for the normal pelvic fin development. However, this gene has this regulatory element. It's an enhancer that can form the Z-DNA conformation and that can form a Z-DNA. And this leads to repeated loss of this element. So when Z-DNA forms, it increases the chances of double-strand breaks that then are repaired, but this element undergoes increased deletion because it can form Z-DNA. And as a result, there is a repeated loss of this regulatory elements, element in the freshwater fish, because in freshwater fish, it is advantageous to lose this element and for this gene not to be expressed. So it's picked up by selection, but the mechanism here is a parallel evolution, and it's a repeated evolution that occurs again in the freshwater fish. So this changes evolutionary dogma. Usually we think about mutations that they occur rarely, that they are random. Here, the mutation rate is high, and also the mutations repeatedly occur at the same site. And non-B DNA can enable such unusual evolutionary mechanisms. So I want to talk about our other project related to non-B DNA, and we wanted to ask a question whether non-B DNA evolves under selection. And remember that we think about non-B DNA as a blessing and a curse. So what about the blessing part? If non-B DNA is important for function, then we expect it to evolve under purifying selection. However, this has not been shown previously. And we looked at this question for G quadruplices, and for them, we had several hypotheses. If they are functional, let's say within promoters, then they should be enriched in promoters. They also should be more stable there, and we should observe signatures of purifying selection for G quadruplices located in promoters. But if they are not functional, let's say maybe within exons they are not functional, they should be depleted, they should perhaps be selected against, and they should not have a high stability. This was our hypothesis. So now let's take a look at what the data show us. So we first looked at enrichment of G quadruplices in different genic regions. So here we're looking at upstream regions, promoters validated in the lab, CpG islands, UTRs, exons, introns, 3' UTRs, downstream regions. Then we also, and in fact, for all the transcribed regions, we are analyzing 2 strands separately. So we're looking at the strand that is transcribed and at the strand that is not transcribed. Then we also have this part of the genome that we frequently use as the neutrally evolving control, non-coding, non-repetitive genome. And then we're also looking at the representation of G quadruplices genome-wide. And then we're looking at the fold differences in the frequency of G quadruplices as compared to genome-wide. And we do this without correction for G content and with correction for G content. So if you are conservative and you want to look at the blue bars, you're welcome to do so. Here we correct for G content. Some parts of this figure, they don't need a statistical test because, for example, in promoters and also in CpG islands, G quadruplices are present 4 to 7 times more often than is average for genome-wide. They're also enriched in UTRs, but they appear to be underrepresented at the non-transcribed strand of exons. Then we looked at the same question, but outside of genic regions, because we know that G quadruplices might also be important outside of genes. And here we are finding their overrepresentation in enhancers and very strong overrepresentation at replication origins. Not so much at recombination hotspots, in fact. That was another hypothesis that non-BDNA might be contributing to recombination, but we are not seeing overrepresentation of G quadruplices at recombination hotspots. Then remember that we can predict stability of G quadruplices, and we can classify them into stable versus unstable. And the next thing that we did is we compared the stability of G quadruplices among different genic regions. So the average across the genome is about 19.5. This is the value of stability. And in upstream regions and validated promoters, the stability is significantly higher than it is in genome-wide. However, if you look at the non-transcribed strands of exons, the stability is significantly lower than genome-wide. Also, in the non-coding, non-repetitive regions, stability of G quadruplices is significantly lower than genome-wide. So outside of genes, the stability is elevated at enhancers, at EQTLs, and at replication origins. Yeah. So suggesting again that some functional G quadruplices are enriched in their stability. And the last thing we did is we looked at the signatures of selection at G quadruplices using the Hudson Kreitman Aquadé test or the HKA test. I'm sure many of you are familiar with this test, but the idea here is that this test can identify the footprint of purifying selection, in which there are more polymorphic sites than divergent sites, indicate sequence conservation. And what we did here is that we compared the ratio of diversity to divergence for non-bead DNA motifs versus surrounding regions, okay? So, for example, we compared G quadruplices within enhancers versus in the rest of the enhancer sequence, just to keep the functional category the same. And what we're finding is that indeed there is selection, which is measured by the odds ratio at G quadruplices, and it's particularly strong for G quadruplices located in upstream regions, also in enhancers, in replication origins, and in UTRs. But again, we have this outlier. We have exons, strands that are non-transcribed, that are not showing signature of selection at G quadruplices. And when we divide G quadruplices into stable versus unstable, so the unstable here are in yellow and stable are in blue, we mostly are observing selection at stable G quadruplices, okay? However, selection doesn't seem to manifest for the stable G quadruplices at non-transcribed strands of exons. An interesting result, and that's something that we are still trying to investigate, is that even in the non-coding, non-repetitive regions, we see in some signature of selection at G quadruplices. And one possibility is that they might still be fulfilling some functions there, and we just don't know what these functions are. One possibility is that they are assisting in a nucleosome array organization across the genome. This was suggested by other papers. So just to summarize this part of my presentation, functional G quadruplices are overrepresented. So they are present more than genome-wide. They are more thermodynamically stable, and indeed they evolve under selection in certain regions of the genomes. And the examples of these regions are enhancers, promoters, replication origins. But exons of genes are the areas in which G quadruplices are likely to be avoided. They do not undergo purifying selection there. They're usually unstable, they are underrepresented, and we hypothesize that they might be costly, they might be detrimental to exons, mostly at the level of mRNA, because the non-transcribed strand later becomes the mRNA strand, and so they might be detrimental at the level of translation. And if you're interested in this work, this was just recently published in Genome Research again. And so you're welcome to take a look at this publication. One thing that kept coming up is whether sequencing error rates are elevated at non-bead DNA, even outside of PacBio technology, because this then can interfere with our studies of diversity, of our studies of selection. And we looked at different technologies, and we can evaluate the contribution of sequencing errors to non-bead DNA. So we used the same data set, the genome in the bottle data set, which is available, publicly available. So we used an individual that was sequenced with 3 different technologies, with PacBio Hi5, with Illumina at 100x, and also with the Oxford nanopore technology. And then we wanted to compare the error rates of the technologies at non-bead DNA versus at controls. So we created two different data sets. The first one used moderate filtering. So it excluded overlapping motifs, non-BDNA of the same type, and also required high mappability. But then we also had a data set with a stringent filtering, in which we excluded motifs overlapping with microsatellites, transposable elements, and also we filtered for quality. So moderate filtering includes all of these repeats, which frequently form non-BDNA. Stringent filtering is the data set, which is much smaller, but it's much more accurate. Then we also used controls, in which we had matched motifs, and they matched the experimental data sets with in number and in length. And then we also removed all biological variants annotated by genome in the bottle. And what we have analyzed are the three types of errors: just the single nucleotide mismatch errors, insertions, and deletions. So I'm showing you the results here for mismatch errors first. And this is how the data look like. So here, the controls are in blue, and the motifs are in red. And these are the distributions for Illumina, for Hi-Fi, for Oxford Nanopore. And this is for moderate filtering, and this is for stringent filtering. The summary is present here, and these are the fold differences at non-BDNA motifs versus controls. And for stringent filtering, for Illumina, there are some significant differences for non-BDNA versus motifs, particularly for G4 motifs and for zDNA. For Hi-Fi, PacBio, so this is another mode of PacBio sequencing in which there are multiple rounds going around the template, leading to very low error rates. And for this mode of PacBio sequencing, we're not observing any significant differences, in fact, in the error frequency between non-BDNA and controls. So this might be technology to go to when one wants to sequence non-BDNA accurately. Yeah. So the other technology, the other error types that we were interested in investigating are deletion errors. And this also are affected by non-BDNA quite substantially. With stringent filtering, direct repeats and G quadruplices are elevated in Illumina sequencing. Hi-Fi PacBio is also affected by the presence of these motifs. Insertion errors as well, even for stringent filtering, especially in Illumina, might be affected by the presence of non-BDNA. So we are still working on this. We have a preprint on bioRxiv about this work, and we believe that non-BDNA should be considered carefully when one is designing a sequencing study, especially if the sequencing study has low depth. And to illustrate this, we came up with the approach to predict the number of false positive SNPs if you just sequence one individual, depending on read depth and depending on the technology. So here the colors are black is all non-BDNA motifs, green is BDNA of equivalent length, and for Illumina, the all non-BDNA motifs are bringing in more false positives as compared to BDNA of equivalent length. However, at read depth of about 17x or so, these concerns are largely alleviated. Okay, so mostly the problem is when you are at the low frequency, at the low depth of sequencing. And this is the case of ancient DNA and studies like this. Okay, Hi-Fi PacBio sequencing is, again, if you reach a certain depth, you are in good position to avoid most sequencing errors. For ONT, it's problematic even up to about 30x because of the overall high sequencing error rate. So Illumina appears to be affected by non-BD DNA more than other technologies. PacBio HEV5 might be the method of choice because of low bias and low error rates. ONT has low bias at non-BD DNA motif, but high error rates. And the general advice that we can give now is that to be able to sequence non-BD DNA accurately, one might want to combine variant calling from multiple sequence technologies because they all have different biases. And then the overlap of the technologies will lead to the most accurate results. So I'm almost done with my presentation. I wanted to acknowledge several people who contributed to this work. And this includes Wilfried Giblet, who actually did most of the work described in my seminar. The first 3 studies were his PhD dissertation. Matthias Weissensteiner worked on the ERRORS project, and we're about to submit it for publication. Marty helped a lot with the statistical analysis. Monica helped with the errors analysis. And then my collaborators at Penn State, Francesca Chiaramonte, Yefei Huang, Kristen Eckert, and Bob Harris. So this is our funding from NIH and also from other agencies. And I would like to use this opportunity to consider, for you guys to consider to work with us on some of these exciting projects. I'm looking for a postdoc to join my group at Penn State. So this is where we are located. We're close to New York City, Philadelphia, Washington, DC. And we are in a college town, which has an unparalleled beauty. So my son took this photograph during the pandemic. So yeah, please let me know if you're interested. Thank you very much for your attention, and I'll be happy to take any questions.